Hey guys, welcome back to One Stop Biology. So in today's video, we are going to study the very last topic of molecular basis of inheritance, which is the second unit of genetics in class 12th, right? So we are going to cover DNA fingerprinting. Now guys, before we start off with the topic, I would just request you to like the video, to share it with your friends, and if you are new to the channel, please do subscribe to the channel. And yes, your feedback is really important. It's very, very important to me. So please do not forget to give your feedback over comments, either if you like the video or if you have any doubt or if you have any specific feedback with respect to the video. Your feedback will basically make me transform the videos better. It'll help me to improvise the videos better as per your convenience. Thanks guys. So let's start with DNA fingerprinting, right? Now, as we said in the last video we studied that once the human genome was completely coded, after the Human Genome Project, we realized that 99.9, guys, remember 99.9% of base sequences among humans is the same. Now, say that assuming that 3 into 10 to the power 9 base pairs are there, right? So out of these, the difference in sequence of DNA, which makes every individual unique in their phenotypic appearance, is very, very less, right? So, if one aims to find out genetic differences between two individuals or among individuals of a population, sequencing the DNA every time would be a very expensive and a very tedious task, right? What I'll have to do is basically trying to compare two sets of 3 into 10 to the power 6 base pairs, and every time we'll have to basically, you know, code for the or identify the entire sequence, right? So what we would do, rather than, there was a method devised, which was DNA fingerprinting. And DNA fingerprinting is a very quick way to compare DNA sequences of any two individuals, right? Now what happens in DNA fingerprinting and what it is exactly? So DNA fingerprinting involves identifying differences, remember, identifying differences in some specific regions or in DNA. These sequences are known as repetitive DNA, right? These regions in DNA sequences are known as repetitive DNA because in these sequences, a small stretch of DNA is repeated many times, right? Now, these repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. So you basically centrifuge, and during this centrifugation, you find different peaks, right? Now, what happens is the bulk DNA forms a major peak, and then there are smaller peaks which are basically known as satellite DNA, right? Now, depending on base composition, which is like whether it is AT rich, AT rich, or GC rich. So based on this composition, length of the segment, number of repetitive units, satellite DNA, right? It is classified into many categories such as microsatellites, mini satellites, etc. Right? Now these sequences normally do not code for any protein, right? In the last video, we also studied that repetitive DNA do not code for any protein, but then they form a large portion of human genome, right? Now, these sequence would basically show high degree of polymorphism and form the basis of DNA fingerprinting. Now, since DNA from every tissue, say blood, hair follicle, skin, bone, saliva, sperm, right, from an individual, show the same degree of polymorphism. So, you take it from any tissue, you will find the same degree of polymorphism, and they become very useful identification tool in forensic applications as well. So remember, such kind of evaluation is very important in forensic sciences, where basically you solve a lot of crimes through this, right? So basically, say polymorphism are inheritable. Now it is inherited from parents to children. So on the basis of DNA fingerprinting, you can do a paternity test, right? So if say if there is a dispute and all of a sudden a kid comes out in the society and says X is my father, and the father who's X says no, this particular kid is not my son. How you can basically, confirm it by doing DNA fingerprinting and doing a paternity test, right? So this polymorphism in DNA base is the basis of genetic mapping of human genome as well as DNA fingerprinting. So basically what we're doing is we are, studying the repetitive DNA part, the non-coding part, DNA part, right? Now polymorphism arises due to mutation as well, right? So basically new mutations may arise in an individual. It can happen in somatic cells, it can happen in germ cells as well, right? So if a germ cell mutation happens, right, it does not seriously impair individuals' ability to have offspring who can transmit the mutation. It can basically spread to the other members of the population through sexual reproduction, right? Now what happens that the allele sequence variation has been discovered as a DNA polymorphism. What? The allelic sequence variation. Right. So what happens now that, you know, if more than one variant, if more than one allele at a locus occurs in human population with a frequency greater than 0.01, it is DNA polymorphism. Say in simpler terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism, right? Now, the probability of such variation to be observed in the non-coding DNA sequence would be higher, right? So, the probability of such variation to be observed in non-coding DNA sequence would be higher, as mutation in these sequences may not have any immediate effect or impact on the individual's reproductive ability, because these are non-coding DNA part, right? Now, these mutations keep on accumulating generations after generations. And these mutations form one of the basis of polymorphism or variability in individuals, right? Now, there is a variety of different types of polymorphism ranging from single nucleotide change to a very large scale change. And evolution and speciation, the entire evolution, such kind of polymorphism play a very important role, right, in the process of evolution. Now let's see what happens in DNA fingerprinting. What is the technique in DNA fingerprinting? So the technique of DNA fingerprinting was developed by Alec Jeffrey. It was developed by Alec Jeffrey. And what he did was he used a satellite DNA as probe that shows very high degree of polymorphism. These satellite DNAs were known as VNTR, variable number of tandem repeats, right? Variable number of tandem repeats. Now, this technique involves southern blot hybridization. This technique involved southern blot hybridization using radio labeled. So we used VNTR, which was radio labeled as a probe. Now, how it happens? So it includes first isolation of DNA. First, what we do is we isolate the DNA, right? Then what we do is we digest this DNA by restriction endonucleases. Digestion of DNA by restriction endonucleases. And then we separate DNA fragments by electrophoresis. Now, after separation of DNA fragments, we basically transfer the separated DNA fragments to synthetic membranes, such as say nitrocellulose or nylon, right? And then we label these DNA using labeled VNTR probe. So the VNTR that we have, the satellite, DNA that we have, radio labeled one, we hybridize it with the DNA that we have isolated. And then we detect this hybridized DNA fragments by autoradiography, right? So if you see this diagram as well, this is DNA fingerprinting, right? Now say we isolate a DNA. So these are basically two individuals, right? These are two individuals. This is individual A, this is individual B. And there is a DNA from crime scene, and we have a DNA from crime scene already, right? Now, basically, there will be two strands. One will be paternal from the father. So one will be paternal chromosome, and the other one will be from the mother. So maternal chromosome in both the cases, right? Now, what we basically found here. So here we saw this 0 to 11. We have basically number of short tandem repeats. So these are the VNTR for us. This is the VNTR from us, from basically the chromosome sequence that we found from the crime scene. Here we saw that there is it is on chromosome 7, on chromosome 2 and chromosome 16. Right? Here again, what we did is we isolated DNA, digested it, separated it, and then transferred it to a synthetic membrane and hybridized it with this labeled VNTR probe, right? So again, this is chromosome 7, this is chromosome 2, this is chromosome 16 of individual A, and this is chromosome 7, chromosome 2, and chromosome 16 of individual B. After this, what we did is by autoradiography, we basically detected the hybridized DNA sequence, the extracted one, isolated one. So basically, we amplified repeats separated by size on a gel. On a gel, this gives the DNA fingerprinting. So guys, now here if you see that the DNA isolated from individual B is giving the same scheme as what is found on the crime scene. Here you have one, here you have one, then here. So both have these. Again here this match, and this match, right? While in the case of individual A, there is one here, but there are no, sizes here. Here there's extra. Here again there are two. There are no here, right? So it exactly matches. This one exactly matches with the one found on the crime scene, right? So that is how we solved the case that the individual B has done the crime, right? And guys, I'll tell you that this is a valid investigation, right? Now this VNTR belongs to a class of satellite DNA. These are known as mini satellites, right? Now, a small DNA sequence is arranged randomly in many copy numbers. The copy number varies from chromosome to chromosome in the individual. So you have chromosomes 7, 2, and 16 here, right? The number of repeats show very high degree of polymorphism. So as a result of size of VNTR varies from size 0.1 to 20 kb. The size varies from zero point 0.1 to 20 kb, right? Now, consequently, after hybridization with VNTR probe, the autoradiogram gives many bands of different sizes. These bands basically give a characteristic pattern for an individual DNA. So here there's one pattern, here there's one pattern, right? Now what happens? These patterns basically differ from individual to individual in a population, except in the case of monozygote or identical twins. So again, guys, if both these DNAs would have been of identical twins, then there would be a very higher chance that both of these would have been the same, right? Now, the sensitivity of the technique has been increased by using the polymerase chain reaction by PCR, and we'll study it later. Now, consequently, what happens is that DNA from a single cell is enough to perform DNA fingerprinting analysis. And by this size, which is basically derived after the autoradiograph, you check it with the one which is present on the crime scene, or which is basically served, which is taken as a sample, like to which you match, right? Now, basically, in addition to application in forensic science, there are many more application where you determine, diversities, where you determine population diversities or genetic diversities, right? And many different probes are used to generate fingerprints, right? So, guys, with this, we have finished the very last topic, which is DNA fingerprinting, and we have completed the chapter molecular basis of inheritance, right? So, we'll be starting with a new unit from the next video onwards. I hope you understood this video. Please do not forget to like it if you did, and share it with your friends. And if you are new to the channel, please feel free to subscribe to the channel for all these updates. And yes, guys, if you have any doubt in the topic of DNA fingerprinting, please do not hesitate in asking questions. You can ask the questions either by posting it in the comment section or texting me on WhatsApp. WhatsApp number is given in the description, guys. But yes, please do give your feedback so that I can improve on the videos even better, and I can make these videos even better for you to understand. Thanks, guys. Bye bye.